Ivy, could you unmute yourself? Ivy, could you unmute yourself? Oh, no.
How
Yes,
about
now it's good. Now it's good, yes.
Okay. Right. So we were talking about the bulk RNA-seq pipeline. So why do we need a pipeline? So the issue is if we have a laptop, we want to see, let's say I want to line 10 samples, not a lot. We have 5 replicate for control, 5 replicate for treatment, whatever. This 10 sample, usually you're dealing with, let's say, 100 GB of data. You want to line them, you want to process them, align them, and then get to your recon table. Do you have an idea how long that would take on a laptop? Usually it takes about 1 or 2 days, depending on how efficient you are. With the pipeline, because we have the cluster, a lot of computation resources, we can basically get it done within 2 hours, regardless of how many samples you are, because we have basically unlimited resources, so you can use multiple cores, so you can analyze them all together. So that's about the efficiency we're looking at here. So hopefully that can be useful for your analysis. If you're getting a lot of samples, we have dealt with 1,000 samples sometimes in this lab. And then for Thursday, we want to kind of go into okay, there are a lot of resources online. There are a lot of public data. How do we find them? How do we download them? How do we actually feed them through the whole pipeline so we can actually analyze a lot of data without even doing any experiment? And then we want to visualize the data a little bit, especially if data are collected in a sequential manner. You want to actually align them together so you can visualize them in a controlled manner so it's comparable, like what we are laying out here is actually a circadian rhythm data in terms of RNA-seq analysis. Okay, so we have, for those who are taking this workshop for classes, we have homework and quizzes. So it's already available, included in the online material. So it's due next Tuesday. So you can get started anytime. But the idea is that by Wednesday, you should have an idea about how to complete everything. So that's like the layout for the workshop. Okay, so let's get started into the first part. We want to have an idea why do we care about bulk RNA-seq analysis at all? Why do we even do it? So let's start with the very basics, biology 101. We want to understand gene regulation, how does the cell control its activities, its functions. So we kind of start from, the mid-50s, when Watson and Crick discovered the structure, of the double helix of DNA, they figured out, oh, maybe there is a mechanism how they can regulate gene expression, right? Crick proposed the central dogma. Basically, it entails that DNA, stores all the genetic sequences. When it's translated, when it's transcribed into the mRNA, which is a species now can get translocated into the cytosol, which will be used as a template for translation, producing specific protein sequences. But is that everything? Is that, everything that we know about gene regulation or how the cell is regulating itself? It turns out it's only part of the picture. Even before that, we know that there are different, transmutable elements in the genome. So that's Barbara McClintock. She found out in the 50s, actually, a couple of years before Watson and Crick got the Nobel Prize and, proposed central dogma. We know that RNA sequences can actually go back and add a DNA sequence. So that means that, there are kind of a layer of feedback instead of one-way street. Actually, there are elements changing genetic sequences. And then another example was later in the 60s was lac-operon, where we start to understand how this regulation actually all works. So lac-operon encodes for one of this enzyme, which is an enzyme that is letting the lactose into the E. coli cell membrane. So what happens is usually there are, bacteria that use glucose. However, in the case of when glucose is not available, they would switch to use lactose as an energy source. So to do that, now the cell, the E. coli bacteria have to upregulate this protein so that lactate can, go into the cell. So it's an energy kind of demanding process. So the cell has to be really savvy about how much protein they produce, right? So the whole process is basically engaged in a negative feedback loop. And this is how that works, is that we know when there are this like complicated picture, but you do need lactose to activate gene expression. So that's the point I'm getting to. So here lactose have to bind to the operator so the repressor can, leave, and the RNA polymerase can bind to activate transcription. So this is another layer of regulation that we, nowadays seems to be treating as a standard. What it does is the protein actually have to come regulate DNA, transcription. So that means that protein is another layer of regulation besides, one-way street where DNA, information passes from DNA to mRNA to protein. And of course, today, the regulation of gene expression in mammalian cells is much more complicated than the bacteria. We have a lot of different complexes. So proteins have to come together to activate transcription. And then mRNA have to, get into the cytosol, which is a rather complicated process. Along the way, there are splicing, there are modifications, you have to trim your poly A tails, etc. And of course, there are different pathways, right? We have signals coming down from the membrane, such as, the MPK pathway. Well, we have a transcription factor translocating to the nucleus to activate specific target gene expressions. So we want to say that, gene expression is everything, but actually gene expression is switched on by the binding of ligand on the membrane. So that's another kind of complex scenario in mammalian gene expression regulation. And then, of course, there are many pathways that actually come together. So study even more complicated story. Or we have, besides them, the AMPK pathways, we have others, such as, the cyclic AMP, which actually converge. So the end result of gene expression is no longer kind of a linear relationship. So once we have the signal coming to the membrane, we will have a, guaranteed outcome of target gene expression. It's like, no. So just want to kind of give us an overview. So forget about the complexity. So we want to kind of step, back a little bit to look at how does it actually work. So we have been talking about the central dogma where, information flows from gene to proteins, and then maybe will, regulate cell behavior and then contribute to organismal behavior. However, we know that there are different feedbacks, especially protein, regulating gene expression. And then, of course, we have for organism regulating cell behaviors. And then a higher level, let's say different seasons, we have different hormonal profiles. So that kind of a higher level control gene expression is definitely something that, we are getting to in the 21st century. So getting back to mRNA, of course, is widely used in modern biological research nowadays. So there are some kind of theoretical foundations of why a lot of people, a lot of us are actually looking at it. And it started by looking at the process of embryonic development, where we, everybody kind of start as a zygote state, and differentiation happens to give us, give rise to a number of different tissues, which, have stark, morphological differences. And maybe they have different composition in protein, mRNA, and even like epigenetic landscape. So the question here is, can we actually somehow make sense of this process? We start with the isogenic clone. How does, different tissues end up behaving so differently? And of course, they all work all together. So the kind of theoretical background of why we're looking at mRNA is that maybe we can unify the expression of different phenotype, trace it back to how they're regulating their, epigenetic network or the epigenetic landscape. So the idea can be complicated and controversial, especially like part of our lab is actually studying that very intensely. So kind of the gist of the story is, if we kind of see different cell types as, occupying a different, kind of diverse landscape, they end up diversifying their gene expression patterns driven by different regulation mechanisms. Might be epigenetic regulation, it can be protein regulation, it can be, different kind of composite effect of different factors. But what they end up doing is occupying different kind of state space that can be kind of, embedded in the mRNA expression space. So that's the foundation of the whole mRNA analysis. I'm not saying that is, 100% true, but that's what most people believe in the field nowadays. And some of us do get that quite ahead, looking at how, mRNA expression levels can be indicative of cell functions, organ functions, or even species differences. So here we are looking at an early study where they characterize gene expression in different organ systems at different developmental time point, and of course, across quite a spectrum of species. What they have done here is a PCA, so it's a principal component plot looking at the first and the second principal component. So this kind of decomposition, I guess you start to appreciate already. So we see this kind of the time trajectory coming down from the center bottom here, spanning out upwards. So that's from early to late, and we can see that different color seems to be segregated. So there are different organ systems. And then different shape indicates different species. And then we can say actually they share a lot of similarities. Doesn't matter which species you're looking at, isn't it? So like on the left, we have green, which is for liver, started in the bottom and then kind of travel upward, and it can be different species. And on the right, we have the central nervous system, we have brain, cerebellum. They seem to actually share quite a lot, even if you're spanning from primate to rodents and the avian species. So that's an example of, in some scenarios, mRNA does give us a lot of insight, and that's what we want to get to today. And another kind of strand of research is very popular is use gene expression as some sort of biomarker. In this case, we're looking at kidney injury. So we can sequence the mRNA species in the same mouse model at different time point of treatment. So if we induce a ischemia reperfusion injury, we can trace how their gene expression in the kidney changes over time. So as we're kind of going from left to right, that's basically indicating disease progression. And then each row here represents an individual gene. And you can see that, for the selected gene panels, they do see a transition from early on normal healthy condition to later, kind of injured kidney state. And then especially for late stage, there are specific genes that are, highly upregulated, maybe indicative of their, injury status that fail to repair. So this is just another example. Is very popular kind of using mRNA as a way to find specific biomarkers that can be kind of a proxy to indicate how well the organ, this cell of interest is doing, and what can we infer about what they're up to. For example, here, a lot of the upregulated gene, the late response to injury, are immune related genes such as NF-κB and STATs pathway. So for my study, looking at the sex differences in the kidney, we actually did a whole kidney bulk RNA-seq experiment at different developmental stage. Oh, let's see. At different developmental stage of black 6 mice. So when we kind of compare them together, and you start to see that for, from the early on stage, we have the PC plot where the time basically spans from left to right. And then the sex are segregated around week 4, which is the time when puberty kind of onset. And we had a series of, perturbation experiment because we want to find out, oh, maybe it's the hormone that is driving this divergence. So we can remove gonad before puberty. We can knock out the, sex hormone receptors at embryonic stage. And we can even kind of validate by, adding specific hormones later on to the gonad removed animals to see, can we actually, at least partially rescue the phenotype. So here, another PCA indicates that we do see the changes due to this perturbation. So we have wild type male and female on the bottom here in gray. So females on the left, the males are on the right. So what we see is that if we remove testes, or knock out the androgen receptor, the male white type now travels all the way to the left. It's pretty much, 80%, quote 80% feminized animals. And in another scenario where we added back testosterone back on, we see that now it's kind of shifted, to the right, which is like closer to the male white type. And then in contrast, the female doesn't seem to bother them much. Like you remove their ovary or you remove their estrogen and estrogen receptor, they don't really seem to change that much in their gene expressions. Only shift a little bit when you add testosterone later on. So this is actually really, I would say very elegant example of using bulk RNA to actually find out the potential regulatory mechanisms of certain phenotype you're interested in. So mRNA, bulk RNA sequence analysis can be quite useful for kind of expedient way of looking at this problems. So before we get into the pipeline, I want to talk a little bit about limitations of looking at RNA data and how we can kind of, bear that in mind so that we don't kind of fall into the traps of a lot of, data analysis. So one thing that we deal with every day is we want to find, phenotypic change, right? We have transition from normal to disease state, or in the kidney example, we have a divergence in phenotype in male and female. And then, we could even look at other transitions. So those transitions it's more kind of step-like behavior. And for mRNA analysis, it's really hard to capture the transition itself because we are only kind of capturing the, what people call the steady state readout, and it's only a snapshot. So that's something we have to bear in mind all the time. And another kind of major issue that we're facing, I felt it in the field, is that a lot of gene expression profiles are highly dynamic. So here we're looking at circadian clock, like period, BMA1. So what happens is throughout the day, their expression level is never constant. They fluctuate all the time. So what happens is that if we do, let's say we do a bulk RNA-seq analysis or single cell RNA-seq analysis, what we end up seeing is actually a very limited snapshot. You have no idea, like maybe we're capturing this mRNA when they're at the peak, or we might actually capturing at the trough or somewhere in between. So if we know that, gene expression that we are interested in are highly dynamic, a bulk RNA-seq will be very limited in terms of what kind of information they're capturing, and if needed, you might want to kind of capture a different time point in a sequential manner. And then another fact, kind of highly dynamic, and the question is, can we even do inference, right? The kind of ideal scenario is when something is up, the other thing is either up or down. And that's the best we can do. However, if they're engaged in very complex feedback, I feel like, a snapshot data would never really give us any meaningful information. So for that, we might want to kind of think about maybe they're smarter, maybe we should do live cell imaging tracking the expression over real time, something like that. And then another thing I want to mention is the relationship between mRNA to protein is highly nonlinear. So we have maybe heard about RNA velocities, which is the idea that, starting from the DNA sequence to get to mRNA species and then get to proteins, it actually goes through a number of kind of modifications. So what happens is there are a lot of kind of connected relationships. For example, we have transcription process, splicing process, or we can write very simple differential equation to capture the relationship. And you start to see that they are actually highly nonlinear. It's not like, one's going to lead to one, like one mRNA is going to give you one spliced mRNA and then one copy of protein. Rather, they're related in a dynamic relationship that depending on the specific kinetics of how stable the mRNA species are and how stable the protein species are. And then another thing that we want to kind of bear in mind is there could be time delays, because a lot of, those processes take time, right? If we are trying to reconcile the relationship between mRNA and the protein, we should actually think about how long does it take to transcribe, how long does it take to modify the mRNA, and how long does it actually lead to protein itself. So here, there was a seminal study actually trying to quantify this process, where they were looking at a frog embryo, and then they tracked the mRNA and the protein species simultaneously in each time point. So what they found out is the correlation is basically very little. If you just compare the same mRNA species and its specific proteins, the median correlation is 0.24. And we know that it's really a poor kind of correlation to deal with. And you can zoom into specific species, where here we have mRNA in blue and protein in green here. And you can see that it's very protein specific. Some of them show high concordance over time, and the others are like, oh, somehow mRNA comes before and protein accumulate over time. And then for the others, it's the other way around. The protein actually steadily decrease. And then this one, mRNA actually goes down, but protein steadily increase. And then the other cases, protein doesn't change at all. Like, mRNA is going all over the place, but protein doesn't seem to bother. So it's not like kind of trying to discourage everyone, but there are caveats trying to draw conclusions and extrapolate based on mRNA studies to infer potential protein changes. So just bear in mind that there are a lot of gaps in terms of delay dynamics and whether it changes at all. So with that, I would like to conclude the introduction part. I have to take any questions, and we can take a break if anybody needs a minute to digest. Okay, we can move on. Just give us a shout out if you want a bathroom break, because we're kind of getting to like 15-minute threshold. So let's go into RNA-seq analysis. So let's first try to understand what is the RNA-seq experiment. So we have, I guess a lot of you have gone through some of the experiment yourself, so we kind of fly through this part quickly. So we start with the whole tissue, or we want to homogenize it to be single cell, or we want to sort them first through FACS. But anyway, we end up having a pool of cells, and we want to digest them. We want to smash them to get the mRNAs out. And then maybe we want to capture some of the species, such as, mRNA with the poly-A tail. So that's the kind of one step enrichment. You can, a lot of us actually do it. And then for those enriched mRNA species, we want to convert it to cDNAs and then amplify them, send it out for high throughput sequencing. So one thing I want to kind of point out is there are different mRNA or RNA species, and a lot of them are what we call it ribosomal related RNAs. And they're actually over 9% of all the RNAs that we recover from the samples. And then we are, I guess, like mostly concerned about protein coding RNAs. If we enrich for poly-A tails, that's what we will end up getting. But there are other regulatory mRNAs that the specific labs were interested in, and they will use kind of specific enrichment process to amplify their signal. And then for sequencing parameters, we want to select the service depending on how, what type of readout you want. The early days, we use a lot of the short reads, which is about like 100 base pair. So for a long chain of sequence, you only read 100 base pair. But if you read a lot of them, you end up, being able to assemble them in the genome. But nowadays, we are dealing with, I think, can be pair-ended 150 base pair. I think they're fairly cheap nowadays. I don't know, maybe you have some experience doing sequencing yourself. I've dealt with 50 base pair single-ended sequencing, you know, those are the early days, maybe 2018. And then, you know, by the time we get to 2020, it's over 150 base pair and the sequencing fairly cheap. I think it's around like $20 per sample, something like that. So if you want to, you know, budget for your experiment, that's, you know, I don't think it's going to be a major concern nowadays. And then, of course, there are other sequencing tools such as PacBio or Nanopore, which, you know, give you long read sequences, which is ideal for primary genome assemblies. If you're working with non-standard species, that might be much more ideal, because if you don't have a reference sequence, it's really hard to, you know, do the assembly yourself. But with the long read, it's fairly straightforward. Like, you know, statistically, computationally, that's no longer a concern. Okay. So what happens in those machines? So here is a quick video that we can watch all together.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
Okay, so that's about what happens when we do pair-ended sequencing, and you get an idea, how we're actually trying to recover both sides of it. And what we want to do is, when you do have the read back for pair-ended, it's actually 2 files, and then we're gonna need both of them basically for the alignment. Okay, so let's go back. Okay. So we have seen like the kind of the mechanism how we do sequencing, but it's like less of our concern. But when we do get the data back, we want to understand, what are in there. They mentioned indexes. Is that something we should care about? Or, if it's pair-ended or single-ended, we'll have to do the alignment slightly differently. Okay, so let's have a look at the sequencing read. So for a lot of the high throughput nowadays, they're quite standard. And typical RNA sequencing result will be looking at, let's say, around 10 million reads. And some of them are, like around 75 to 150 base pair for each sample. And then this is, the information stored in those FastQ files, which is an efficient way to store like millions of sequences information. And they're typically pretty large. For a human sample or a mouse sample, the raw read actually can be gigabyte large. So if we say that for pair-ended, that's 2 of them. And if we have 10 samples, that's 20, let's say 2 gigabyte per file. So get an idea how big the data set you're dealing with.
So the FastQ, I think you're about to answer that in a minute. Like what's in the
right? So here's just kind of you can actually open the files and have a look at it, but it's pretty boring in my opinion, where they have basically several rows for individual reads, and they have sequencing. Now they have the sequences laid out, TCGA, and then they also give you another row which is for the qualities. So those are encoded in, I would say, non-human readable languages, but for specific softwares, they can handle that for you. And then we have read 1, which is the top 4 rows, and then followed by read 2, so on and so forth. So we have specific IDs, and then the sequences, space line, spacer line, and then per base qualities. So that's the information we need to actually do the filtering quality control step. So we can specify how stringent we want to be. And let's have a look at the base quality. So for each of the base, basically it's a quantification of how confident I am about this base call, right? Because it's all fluorescence based. That comes with, maybe it's noisy. Maybe the flicker didn't, get recorded very well. So what they end up giving out is different scores for individual base. So it's basically a kind of representation by error rate. So for example, a score over 10 will be a rate of 0.1. So that's like I'm 90% confident about how good this base call is. And then we are typically looking at 20. So that's about 99% confidence. So as of course, you can go on and, to be more stringent, but we are going to try the score cutoff of 20, which is a 99% confidence for the base call. So let's have a look at the workflow. So we have seen this earlier. So we want to kind of start with dealing with a sample FastQ file, and then we will do the quality control filtering step today. And then for tomorrow, we're going to continue with alignment and generating recount table. So we're going to be working on Hoffman 2. So it's a super, let's say, a supercomputer is a cluster. Basically, there are hundreds of different machines that are available at your, personal laptop command line console. So let's just have a quick view about how it works before we take a break. So we have many computers, and they're all kind of, connected to each other. So when we log into the command line, we are basically in the login node. So the login node is shared between like all of us. So we're kind of sharing like the portal of the cluster. So what we are doing, let's say we want to run a job, like we want to do a quality control or we want to align the sequences. So what the scheduler is doing is sending the command to an individual node that is kind of working for you. And then once the analysis has been completed, the result will be returned back to the login node, so you can check the result or any of the output. So that's the layout. One thing that I do want to kind of remind everybody is the login node is very vulnerable. Let's not crowd it. For example, if you have any kind of computational intensive task, don't do it in the login node. So what we want to kind of encourage everybody is to request an interactive node. So you have, let's say we have, we request an individual node so that if I'm messing around, with it, nobody would even find out. So that's what we want to kind of establish today. So, everybody will be comfortable working on Hoffman 2, and we know how to request an interactive node. And we want to do simple file manipulations, do quality control, filterings, and then that's about the goal of today. So, on Hoffman 2, there are around 21,000 nodes. So that's about like very big computer. And then, of course, in the email, I mentioned that, we are going to be using Bash, which is an Linux language, to navigate Hoffman 2. And I have a cheat sheet available for everyone, so you can kind of put it on the side in case you don't know what to do the next. And then for a specific task, we're going to try interactive node today. And then for tomorrow, we can try batch jobs. Basically, you can send hundreds of different tasks to the machine, and you don't need to, monitor every single second. And then for different softwares, we want to kind of take a note. So all the softwares we're using in this workshop will be available. But in case in the future you're working on your own and you want to, find out, oh, I need this software, does Hoffman has it? If they have, you can just, kind of use the pre-installed software. And then many labs have their own softwares. So within your group, you can, kind of borrow or use lab shared resources. For many of research purpose, there might need custom built softwares, so you might have to kind of build it yourself. So that is available, I think, with some caution, with permission. If permission is allowed, you can do that. For some of them, it would touch the root access, then that's a problem. And then for the cluster, I just want to mention again that this is optimized for parallel computing. For example, we have hundreds of different bulk RNA-seq samples. We can do it in parallel to, minimize the computation time. Instead of 2 days, we can get it done within hours. So that's about like the idea. And today we want to go through exercise 1, 2, 3. So we can take a break. I think it's a perfect time. So we can stretch a little bit. And then when we come back, we can open up this document, and then we can go through it step by step together. Okay, let's take a bathroom break for five minutes. Good. Okay. I see. So it takes some time. Yeah, yeah. Maybe you can just follow along for now, and then you can catch up. Okay. Yeah, so it's a way kind of a slow pace. Oh, okay. Yeah, should be good. Yeah, we should have a lot of. You can just rush. Okay, sounds good. Thank you. Sorry. Well, anyway, I passed. It was so yeah, it doesn't show. No, I just have to type right now. No, yeah. It's busy. Yeah. Okay, so I will be trying, and let me try to log in. So that's how it looked like when you trying to enter SSH, and then we'll ask for your password. So you type in there, and then hit enter, and that's the home page. So we're here, right? So we logged in successfully. And then we can check where we are. So it says login to. So I think there are around 4 nodes in login. So we can list the content. We have, I have a couple of folders in the home directory. So we can try to request the interactive node. This makes sense. I have it. I'm looking at it. And then if I don't use it, I don't know what it is. No, just it's allergy, so we're getting copy pastes. And those probably going in. Or not. Okay, so most of us are on the cluster. Yeah, so now you have one.
Okay,
Okay,
now I have it.
yeah, for requesting a no, usually take a while.
Okay.
If you wait for too long, let's say more than 10 minutes, I'll say quit, start again. Because the scheduler sometimes is very busy and then they stop responding. And that's bad news. So what I usually just kill the whole thing, start, and then try again. And then for labs with their own node, that's good news. So you have basically reserved capacities for your whole lab. So there are cases where I have to wait for 2 hours for a node at some point during the day. And then the lab node is much faster. So that's about like the options we have on the cluster. So now we are in a intact node. So you can kind of check, because now the name changed. So it says N7155 can be different for you because you might be on different node. So now we're in a separate machine, basically. So the default is you're back on your home directory, and then you want to navigate to the directory you are interested in. So we want to move on to exercise 1B, which is to obtain sample bulk RNA-seq data. Here. So you can make a folder in your home directory, and then we can copy the files into the data folder. So here we are dealing with 2 FastQ samples, which is a bridged version, so they're smaller in size. And they're all from human bulk RNA-seq experiment. So let's try to go through this. Can we successfully copying the data? Okay, so that's, yeah, so I have, the files stored up in the cluster. So another option is to download the file from a Google Drive and try to upload it to Hoffman 2. So this is kind of redundant, but it's basically trying to set us up so we can learn about how to transfer files on and off Hoffman 2. So I use FileZilla. There are different protocols for PC, for example. But I think that's a good one to deal with. So maybe we can do this later at your own time, because we do have the files on the cluster today. So for the homework and the exercise, we'll have to go through this part, but you can just follow the steps here. Okay, so exercise 2, we want to do quality control on the FastQ files that we've just copied. So we have 2 FastQ files, KD1, SC1, and we want to make sure that we know what's in there. So we can view the content, and we can check the number of, how many lines are there. So let's find out how many lines are there for each file. And then have a look at what a read actually looked like. Oh, yeah.
Let me see the layers are such. One is like a CPU and something.
Oh, it's in the data. It's a view list. It's in the data folder.
Oh, okay, so what do we do?
So you CD, you go into data. Folder, CD data.
Oh, CD data.
Yeah, so that's going to the data.
Just CD, right?
CD data. So you want to specify. Yeah,
Okay, so.
and here, so now you're in the data folder, so you can do last.
Oh, we have to go into maybe cheesy because okay. Okay, now here we go.
Yeah, so now you can see the content, right?
Yeah.
So we have V1, V2, etc.
So what's going on here? So we have to go. So what's this doing? That brings us to the data.
Yeah, so you created a folder for data.
Okay.
You copy the FastQ files into the data folder. So all the 2 files are gonna be in the data folder. So if you want to view it, you either go into the folder first and then you should come on last, or you can try a different one. So let's try last. Sorry, you go up, CD. So you go back to the original folder. You can also say last data.
Oh.
So you can also view it, but if you're not in the folder, you have to specify where the file is. Does it make sense?
Yeah,
Like, keep
yeah.
the
I'm not in the, okay, gotcha.
Give it. That's
I'm trying to see what maybe this is.
Oh, you. Oh, there is another data in the data folder. Oh, okay. Right, so that's the folder. It's a different color. And I think what happened is you copy the thing into a folder that doesn't exist when you copied it. So there is a file data. So what you want to do is to go in there, delete it. No. So there the file dependencies. It's very strange in here. So you delete this thing and then you copy it. So now you have a folder. So you can copy.
Question about the last minute, how do you return to that?
Oh, Q. Press the letter Q. Yes, to quit. Less.
Okay.
I want to see both of them. Can you see them like together, or did they get out, right? To see the next one, maybe one. FastQ GZ WC.
Right, yeah, you have to
Oh,
quit
that's-
first.
never mind,
Mhm,
sorry.
yeah. Yeah, we can try again. We'll try together.
What,
Oh, so you can create the aliases that, customize. So S for me is less in the long view, but yeah. It's just like, because I use that a lot. Instead of typing 5 letters, I can type 1 for all. Okay, so let's have a look at the KD.
you keep, I didn't.
Yeah, you can create aliases as you like. You let's say like, yo, I want to, find out what I typed before. It's like history, instead of the whole word history, you can just say H for the history, things like that. Okay, so that's the file. I hope we have seen it already. So we quit the viewer by pressing the letter Q. So we can check how many lines there are.
So that tells you how many lines the file has,
Mhm.
but it doesn't tell you how many of this, the
read.
there.
Yeah, we gonna do some math. 4 lines a read. Does it make sense?
Oh, every read is exactly.
Yes, 4 lines of code. Yeah, 4 lines of contents, right? Yeah, so that's
Even
what
if
it.
the read is long, like it counts as a single line.
Yeah. Well, it's not that long if you have the other day I actually asked like people count. Have you tried to figure out how long the read is?
Well, you mentioned there were short reads and long reads. Earlier.
But in a single file, they should be the same length. Right? Because you did a specific machine. You did a specific library prep, specific machine for sequencing, and they should be the same length.
Got it.
Can be 50, 100, 150. So the files we're dealing with, it's 75 base pair. We have,
Divide by 4.
yeah, so we have 75 in total here. So basically this file is actually very well formatted. It cannot, kind of continue. It doesn't vary.
I see.
Yeah, so it's very regular.
The N is not a field type that they are certain about.
Yeah, I think that's the index
Oh,
in the beginning.
but the rest of it is the.
Okay. Are we good with this part? Yeah, we can try to read out the score, but that's like the 4th line.
If you do top head.
Yeah,
the
if
first
we do
10,
head, yeah, it'll be the
we
first
set
10.
portion of
Yeah,
it.
we can try that. Let's try to do head. KD. It's gibberish. Because it's a gzipped file. Right, right. So for head or tail, you might have to unzip it first and then it will print out. But let's, kind of let you see without unzipping.
Yeah, but can you show me where it's the previous score index? So let's say this is the first and back.
Right, so that will be the one with the hash.
This one, how do you, so this is the whole thing is the previous score?
That's for, I think, each base. So ladder for each base.
Oh my God, so
Okay.
then how do we,
The call behind has to be like that.
they would, let's hold read of 75.
Well, the big news, we don't have a good memory.
Oh, okay, there's something.
Yeah, so it's coded in human language. So when we are gonna do the quality control, the software take care of it. So you just have to make sure that, we specify how confident we are. So we're using a score of 20, so that's 91%.
But that's downstream after we. breaking that up.
Yeah, so the software takes care of that.
There's no way I could convert this to for 20, 30.
We don't need to, yeah, we don't need to. But all the information is, written in the FastQ file. So that's this idea that we want to check, what is in the file, what information do they contain. And, but you're gonna use the software to find. You're not gonna do it manually.
So tedious.
Right? Exactly. That's not it. But the fourth line basically, the fourth line. The quality or the basic. Okay, so we have gone through 2A. So let's try 2B. So that's using FastQC to do quality control. So FastQC is a software and we have it pre-installed on Hoffman 2. So we can just load the software. So if you're curious to find out what softwares are available, you can type this thing into the command. Let's have a look. It's very slow.
Please.
Okay. So here we go. We have a very long list of softwares. Okay, let's just start from the top. So those are the master ones. And then specific softwares, something that you might recognize already, like Anaconda, the different virtual machines and different languages like Julia language, Python, we have Mathematica, MATLAB, and then there are other things like the GCC, which is for compute kind of building softwares, different versions of R. And to come down, we have cell rangers, those for aligning single cell sequences, if you need to do that. And SAM tools. So that's something will be used tomorrow, handling the BAM files. SRA tools, those will be used on on Thursday. So we'll be using that. We can use that to download public available sequencing data. We can have a look at others. Yeah, so that's the whole list. So we have FastQC pre-installed. Let's see where they are. So it's alphabetic. So here we have FastQC. So that's the one we're loading today. So let's try to load it. Okay, so we load it by doing module load. FastQC. So the version is this. So we load the software. And then we can look it up, like how to use this software. So if you have like help at the bottom, so we'll kind of, load the whole menu basically in case that you need help. But for today, we can just work on the FastQ samples we have. So if we're in the workshop directory, so you can just do this.
We have already unsifted. We still need to enter the .gz or.
Oh yeah, so this is for the original FastQ that is zip, right? So this is in where we put the files in.
Right, but if I unzipped it already in that state.
Oh yeah, then you can forget about it.
No, I haven't done the zip one. Is that what you want?
Hmm?
No, I have a
Zip file.
zip file.
Yeah, that means it ran successfully.
Should I do the other one then?
Yeah, you can try the other one. It's like the SD. Mhm.
I also like the history of them.
Yeah, try that.
Wait, my H doesn't work, so.
Oh, you have to specify the alias in your bash file, bash profile file.
So we're doing this for both KD1 and S.
Yeah.
I also got HTML file.
Yeah, so the HTML, you can transfer it to your laptop, and then that's gonna give you a browser view of the quality control results. So you can try to use FileZilla to download it to your laptop. Oh, yeah.
Yeah, without your data folder, then you want to run. Okay. Just make sure that you know, like, the path is right. Yeah. Sometimes it's like, software is like, where is the file? I'm not sure.
That makes sense. Thank you. Have you transferred files to your local computer?
Right, so that will be going up to exercise 1C here. We use the file transfer protocol. So if you have not set it up, we can kind of try to do it now and then download the HTML output file to your laptop, and then we can see it in your browser.
So this FastQC software that we ran on our post file, what did it exactly do? Right? So we ran the FastQC software on those two files that we had down.
Right.
What did it do?
Oh, so it has output. Can you find out where the output is?
Well, I see the KD1 underscore FastQC zip.
Yeah, that's the output.
Yeah.
And then the HTML file, it contains all the quality assessment of the file, but we have to download it so we can see it on the browser.
Ooh, that's cool. I don't know what I'm looking at, but that's cool.
So we are looking at this div file, right? So that's the output. And then we also have an HTML file. So that's something we have to download to our own laptop. And then you will open up a browser for the readout. So I'm gonna open it up now. But for you, you might want to set up the file transfer first. So let's take some time to get FileZilla to work, because I do want everybody to be able to download and then see it for yourself. Okay.
Oh, so I don't have FileZilla on my computer.
Yeah, we have to download it. Yeah, Sarah got to work too. But you'll have to set up the connection. You have it. Yeah, that one. Let's, yeah. Yeah. Yeah, exactly. Yeah, it did. There you go. Yes, so the FastQC basic thing, all this.
Something is not good.
Yeah, it's really the basic. Yeah, but this is an example. It's right. Okay, so let's set up FileZilla and then download the HTML file.
Find the files, like, because I don't. Shit. These are. Yeah.
Do you have FileZilla?
Oh. When you start. Oh.
Yeah, you have to install it.
Just download
The
the
file, yeah.
okay. All this.
Yeah.
So just download.
So now you have the software, we will need to set up so you can connect to HotNet too.
Put a file site manager.
Use and then
Or
you
just
give it a name.
okay.
And then keep on setup.
What should be faithful towards the connection of the team? I found what it is. So this is the whole. With my.
I think it's a different setup. We go back to
This was administration.
Oh. Maybe we can leave the client, not the server. Yeah. Can you use the link over there? There's no link. So, 5-0 is a Zoom. One second. Yeah, client. It's like, oh, I've never seen that before. Yeah. First one with. I'm being impulsive. Not so weird. You downloaded it. Okay, I think not in your Thomas. Interface. And then you set up the connection.
Oh, the username and password are on my Hotmail to trust there.
Let me find the files on the file so. This is where this is me, my computer. This is that's the bottom here. Okay. Yeah, you go to the bottom. I don't know which to. Beautiful thing. Yeah, try something again. Usually when it's all day, it's graduate. So maybe you have again to somewhere else. So we get it to work. So I have it downloaded and let's have a look. So that's basically the output from FastQC. So on the left are the things that has been checked for you. And there are different categories. And you can kind of go through it in detail. But basically the idea is give you a rough estimate of how good the sequencing went. And usually there is nothing to be really concerned about because when you filter it, you kind of get rid of a lot of the concerns anyway. So especially when you have a lot of samples, I barely really look at the HTML and the ML files. So, but for today, it's more kind of, you do want to check the files are there for you. And you can kind of, keep it as a record, in case you want to revisit later. Okay. So let's go back here.
Is there anything major from the FastQ file if, you're doing the check before you continue with the analysis? Anything major that sticks out that
Mhm.
would say this isn't a good run or something?
Yeah, there are cases I've seen, just went miserable. That means that the sequencing didn't work. Most likely the sample quality was too low. It wasn't like, somehow the sequencing failed. Or rather the cDNA library was not in good condition where it got decayed, got degraded somehow. So that's something you watch out for. Basically, it's like this sample cannot be used, so you have
If
to
it says,
throw it
like,
away.
poor quality reads then at the top of the.
So you would actually see that, this is gonna be a lot of red.
Mm.
Yeah, that means there's something serious wrong about the sample. Yeah, for that case, it's like, the samples cannot be included in the data analysis. So I'll just throw it away. You might want to do, kind of second try. If you have, residual biopsy or tissue left, you can, get another batch of our mRNA, send it out for sequencing again. So that's about.
Do you get differences in reads, I guess, or with different tissue types? Would you get different efficiency for the sequencing or anything like?
Yeah, the depth definitely is gonna vary.
Mhm.
Even from sample to sample can be different.
Mhm.
But it's not really a big concern if we can actually make sure that the quality is good enough. Because in the end, you want to have that quantification, right? The more read, of course, is gonna give you better coverage.
Yeah.
But it's still comparable because we will do kind of on third days we can do normalization to minimize or to somehow control for sample variation. So as long as they didn't fail, it can be used and it can be aggregated, compare even between, across different samples, different organs or different tissues. Okay, so we have gone through quality control. Let's now try filtering the low quality read. So that's exercise 3. So for this one, we'll use another software called Fastp. So Fastp is a very small, portable, executable file. So what we do is we can either download it or copy it from the folder here. So 2 options you can try if you like.
We couldn't just use those module Fastp.
So Fastp is not pre-installed, but it's so small that you can just basically, pass to anybody, you would like to share. So we can just copy it from. Okay, can we get the software all right? Okay, so basically it is one step, but we want to kind of go through like what we are doing here. So we are using this software and we give it the input, which is the zipped FastQ file for KD1. So this is where it is. It's in the data folder. And then what we want to kind of specify the output is also in the data folder with a different name. So I called it PostQC, FastQ, also in zipped format. And then the parameter we're giving in is the length requirement must be above 20. So that means that for 75 base pair sequencing, we want to make sure that all the read that we're processing is above 20. And then the average quality score is 20. So that's 99% confidence in the base call. So that's for every read. And then we want to have a output file, which is HTML, and then another one JSON. So HTML will be the same thing that we have seen earlier. So we can kind of basically using FastQ for quality control as well, if you prefer. So we can just copy and paste the whole thing onto the command line.
Is this permission denied? Wait, do you copy this? It says permission denied.
I think if it's the fact that it does it won't go.
But I thought I downloaded.
You have Fastp. Can you see where it's over?
Well, yeah, I mean, I'm assuming.
Oh, you downloaded. Okay, can I check?
So copy it all. Yeah, and it will go in, but the thing that you don't want that, you don't want the key. So I copy and paste it.
Yeah,
like
you
a
have to
Word
change.
pad or notepad or something.
So when you download it, you have to change the.
You can copy it all of it. It sometimes pops up in the corner. And then I put it into like a notepad. And then do that. Yeah, only because I've got the same issue as you. GZ.
You want screen?
Oh,
So now it's executable.
Mhm.
Right. So just one note for those who has downloaded the software, you want to change the permission to use this software. So A+X means now you can execute this file. If we copied it, it's fine because it's already executable.
Then you should just.
Yeah, try again, Melanie, to see whether that works.
I think it's the. I just don't know how to go to the next. Separate. Yeah, the backslash means it will continue. Is it like the pie? No, it's. I think it's just this one. Yeah.
Is the backslash like pi? Backline? Or if I were to
Yeah, that
enter
one.
these one by one, I had to do first line, then again dot slash Fastp minus O, the same thing. Is that
Yeah.
I had to run it in multiple ways? and
So
multiple lines.
yeah, so for this, just copy and then paste in one go, enter.
That's what we did.
Yeah. So if you're typing, you can just forget about this slash and then just, go through a single line.
But I have to say dot slash Fastp for the second
Yeah.
line. Just repeat the command.
Yeah. So for a single line, we can try to do that. Here. Right, and then you continue with the second line without that backslash. I'm basically copying it from the script. For script perspective, you want to kind of, lay it out so it's easier to see. So if you are just typing it, you can just type it in one line.
don't need the straight pipe.
Oh, you don't, yeah, you don't need the partition. Yep. Okay, can we get this to work? Okay, yeah, some of us has gone through it. And then we can check what output files has been created and does it have any kind of output to the console? Sometimes it says, oh, you know, we filtered X, you know, how many reads.
It says the output to file.
Yep. And then you can also check the post QC. You know, what is in this?
HTML file.
Yeah. Yeah, you can also download this HTML and then check it in your browser.
You can check it.
Mhm.
I only got one HTML file. I didn't get one for like, just the files. Yeah. Yeah, you should write out. It should, right?
Yes.
Sign in. Is it? The part? Maybe I need to what's this? Yeah. You don't.
Oh, somehow he changed this.
Oh, it has a different name. Maybe there's a space.
Did you copy and paste it?
I do.
You go to the,
Oh, this is what page.
yeah,
Because you're.
just, yeah, copy the whole thing. Then you go to the. Yeah, okay, enter.
But just the K one.
Oh, right,
But how do I do that?
So you can use a text editor on your laptop.
Something's putting it. Here.
Oh, yeah, and then you change. That's.
Everything.
Yeah.
Yeah, so basically you have to specify, I don't know, like maybe the space. Yeah. See. Are we all good with this step?
I only got one HTML file for that thing as well.
You get one HTML and one JSON.
Yeah, Jason. Yeah, so for each round, we get to output, and then maybe we should have the post QC file somewhere.
So I see the post PCB.
All right. So you can just do that. Yeah, this is. Oh, enterprise is weird. You can try terminal on that or on Mac. It's way simpler. Yeah, I see that's a bit junky. I'm doing this on my. Yeah, try terminal. Yeah.
What we are doing here is the bigger sample. So I don't know, that seems like I think after the shots. Yeah, we can try this. Yeah, it's from me now. It's almost the same.
All right, so for those who have gone through this part, we can move on to exercise 3B. So
I ran it, it seemed to work, but I can't find the file. I don't know where. Take a while. Right, didn't figure something. And then all these are. That's right. I didn't get to register. I heard you were hosting. Right, you have the HTML. Oh, and QP. Yeah, I should. It says it has a very good HTML and JSON. Okay. Yeah, so if you didn't make it, no, you're gonna be smarter. Yeah, you run it in the home directory, the workshop directory, temporary solution too. Otherwise, you have to specify in the code. Oh my gosh. It's okay, but I don't think it's there. Like I did it in the Russell as well, but it didn't generate anything. Even though it's like, I don't know, it seems to have run fine. Yeah. Maybe you have the same issue. I found a game list file data. If you just use like that to do with this. It looked, I ran it from here. In the workshop.
I think it's written out as Fastp.html. So some of us have seen that. So it's not written out with the title, like this PV. It's just written out as a generic name. I think that's something to do with the space and the backslash.
So that's usually the same thing. Okay, all right.
okay, so for exercise 3B, we're gonna use a pair-ended sequencing read for mouse liver data. So we have the script here. So we copy it to your home directory, to the workshop directory, and then we can check what is in there. And then we'll need to modify the batch script. So you can have your email in there so that when your job is submitted or start running or is finished, you can get notifications. And then basically we do this qsub command to let the job run on the cluster. And then we do that for 2 samples, one wild type, one knockout, one wild type. Okay, so let's try this part. you tried this part yet?
No.
Okay.
We do Q stats. Oh, yeah.
Yeah, so you can check the status.
It already, I just got the email.
Okay, yeah, great. So for modifying the script, we have appendix at the bottom. So basically how to add it in Nano. So the appendix here, like how to open the file and then move around, move the cursor. And then we want to add it and how to save. So Control O to save, Control X to exit. So the basics, yeah, for those who have worked with it before, must be quite straightforward. So yeah, let's try 3B. And that's about all the content that we have today.
One thing.
Hmm?
Where if I put everything on the phone, I'm good to go, right? It's on system.
Yeah, it's running. Yeah, perfect.
Am I good to go?
Mhm. Yeah, so we can see the result tomorrow.
Yeah.
Okay, sounds great.
And tomorrow for the class, is it like because I have another class at 3, so I'm trying to allocate my time so that I can go to both. If I watch, I know I can get the recording from people. If I watch these videos, are they like the same thing or
Oh,
like
those are old, yeah.
these are the same, right?
It's not the same.
Or
Yeah,
Yeah.
those are from 19.
okay, okay.
Is it wrong?
Yeah.
So when origin is 19.
Oh, that's not you.
That's alloy.
Yeah. Okay.
Yeah, so I don't have that slide.
These are not
Oh,
accurate. These are not accurate.
oh, sorry.
These are not accurate. So I should just sit in the corner with you.
I think like when origin taught it, it was a different style.
Oh,
And the
okay.
contents are different. For example, I have a pipeline here. We didn't,
Oh,
didn't
oh.
introduce that.
Alright, then I'll just sit in the corner with you. That's okay.
I see. Okay. Yeah.
Yeah.
So we, yeah.
You have to change. And then.
Okay, so they know what the file they're gonna.
Yeah.
Yeah, so the only thing I have to do is add
Change the.
and leave the little.
Yeah, leave the hash, so control O to save. Oh.
Oh. I'm like, I'm doing all.
Yeah, enter. So we saved it and then control X to X. Yeah, great. So you can check the file again. Yes. Sure that it's right, it's email and then try. See, yeah, I think it's good. And then you can go through the piece out.
Okay, and now I can go to the. Yeah, I just was stuck there. Okay, let's do that.
I got two emails saying the layout was complete.
Okay, yeah, then it's like. The output, so the output is gonna still be on the cluster, so the email is just for no. So we're gonna go back and come on. And then we can see the content. Yeah. Right, so it looks right.
And probably exactly the need to be curious what the exactly did.
Oh, okay, yeah, you can open up the script. You can view it, so if you say last, this screen name. That's for editing, so you go back to the one. And then it's it was manual, it's wrong. See what is in there, what's what it should. Let's do some drawings for John. And then that's what we did basically. We filtered the meetings.
This would be that. So we're gonna still doing.
Yeah, that's
So now. The results. If the results of that filtering are in here. That Jason. I don't believe.
That's good. All right. So I guess
Double get it?
Yeah.
Yeah, no, man, it's me taking it or?
That's good.
Okay, so, I appreciate you to get back to the. But I will work with her to see, well, alternate time, like you said, it's this.
Oh.
Sorry about that.
Okay. Yeah, but I just noticed like the email, you know.
So let's see.
We can track this. This is like on to separate places.
Because in theory, we should submit too.
Oh, it's okay.
Thank you. It's okay. Yeah, it should be fine.
Yeah.
Yeah, it's gonna write out to.
Okay.
To.
That was this.
Right.
And then I checked. And tomorrow.
Yeah, we're gonna go through 4 to 6.
Okay.
Okay, so if we have gone through 3B, then we're pretty much done with today's contents. So we're gonna reconvene tomorrow afternoon, 1:30, same place. And then we're gonna go through the rest of the pipeline and then go through exercise 4 to 6. So with this, we can conclude, and now I'll see you tomorrow. If you have additional questions, you can stick around, and then I'll be able to answer that. Okay, great. So I'll see you guys tomorrow.
Thank you.